All right, let's do this. We're diving headfirst into the microbial world, the invisible universe that's happening all around us and even inside of us right now. This is going to be a fast-paced look at how these tiny organisms tick, from how we fight them to how they power themselves and even copy their own DNA.
So here's the big question that drives everything. How on earth do these microscopic creatures not only survive but thrive, multiply like crazy, and so often stay one step ahead of us? That's what we're going to unpack. We'll start with our side of the story, the fight against them, and then we'll flip the script and look at the brilliant mechanics of how they actually run their own lives.
So here's the game plan. First, we'll cover fighting the invisible enemy. Then we'll peek inside their cellular power plants. After that, we'll see how the replication machine works. And finally, we'll explore how they're flipping the genetic switch to adapt to just about anything.
Okay, let's jump right in with part one. This is where we, as humans, really enter the picture. We're talking about antimicrobial chemotherapy. It's the science and the art of fighting back against these invisible invaders.
First things first, let's get our terms straight. An antimicrobial agent is pretty much any substance that can either kill microbes or just stop them from growing. Now, antibiotics, that's a special class. They're natural products that go after bacteria specifically. You know, this all kicked off with Alexander Fleming's famous accident, where he saw some mold, penicillium, wiping out bacteria on a petri dish. And just like that, the antibiotic age was born.
So, how do we figure out if an antibiotic actually works? Well, we do tests like this one. What you're seeing is a dilution test. See how each row has those little cloudy wells? That's bacterial growth. As you go from left to right, the drug gets weaker and weaker. We're basically looking for the exact point where the cloudiness disappears. That's the sweet spot, the lowest concentration of the drug that can still stop the bacteria in its tracks.
And that sweet spot we were just looking for? It has a name. The minimal inhibitory concentration, or MIC for short. This is the magic number, the critical piece of data that tells doctors and scientists exactly how powerful a drug needs to be to shut down an infection. It's absolutely fundamental.
So, how are these drugs actually doing the dirty work? Well, they've got about 5 main strategies. They can mess with the bacteria's cell wall, jam up its protein factory, the ribosome, block its energy-making pathways, stop it from copying its DNA, or just tear apart its outer membrane. The trick to all this is that these drugs attack things that are unique to the microbe, so they leave our own cells alone. Pretty clever, right?
But here's the thing. It's always a balancing act. You have the therapeutic dose. That's the amount you need to get the job done and kill the infection. But then you have the toxic dose, the point where the drug starts doing more harm than good to you. The bigger the gap between those two numbers, the safer the drug is. That gap is called the therapeutic index. A high index means you've got a lot of wiggle room, which is exactly what you want.
All right, so we've seen how we fight them. Now let's switch gears and look at what makes them go. We're heading into the cellular engine room to talk about metabolism and energy. This is how they power everything they do.
You know, at the heart of it all is this beautiful cycle. On one side, you have photosynthesis, which takes sunlight, carbon dioxide, and water and turns them into sugar, glucose, and the oxygen we breathe. Then, on the other side, cellular respiration takes that sugar and oxygen and burns it to create chemical energy, or ATP. It's this perfect planetary partnership. The waste of one process is the fuel for the other.
Let's break down that cellular respiration part. It happens in 4 main stages. It all kicks off with glycolysis, which is so fundamental, pretty much every living cell on Earth does it. Now, if there's oxygen around, we keep going through the link reaction, into the Krebs cycle, and finally to the grand finale, the electron transport chain. And when I say grand finale, I mean it. That last stage, the electron transport chain, is the real powerhouse. It can crank out a whopping 32 molecules of ATP. ATP is like the dollar bill of the cell. It's the energy currency. The earlier stages only make a couple of ATP. So this final step is why breathing oxygen is such a game changer. It unlocks this incredible amount of energy.
But hey, what happens if there's no oxygen? Well, life finds a way, and that way is called fermentation. It's a plan B for making energy, and its byproducts are things we use every single day. The lactic acid in your yogurt and kimchi, that's fermentation. The ethanol in beer and wine, fermentation. Even the holes in the distinct flavor in Swiss cheese, yep, you guessed it, fermentation.
Okay, so microbes have energy. They're fueled up and ready to go. What's next? making more of themselves. And before any cell can divide, it has to do something incredibly important, make a perfect copy of its genetic instruction manual. Let's look at the DNA replication machine.
It's funny to think about, but we didn't always know DNA was the star of the show. It took a series of landmark experiments to figure it out. It started with Griffith, who noticed some mysterious transforming principle could pass from one bacteria to another. Then Avery, McLeod, and McCarty nailed it down. That principle was DNA. Finally, Hershey and Chase sealed the deal when they showed that viruses infect cells by injecting their DNA. It was a total paradigm shift.
Now, when it comes to copying that DNA, bacteria are built for pure speed. Unlike our cells, with their complicated library of chromosomes locked in a nucleus, bacteria have one single, circular chromosome just floating in the cytoplasm. It's way faster, needs fewer parts, and, get this, they can start making proteins from the new DNA while it's still being copied. It's multitasking at a molecular level, just mind-blowingly efficient.
To pull off this high-speed copy job, bacteria have a whole toolkit of specialized enzymes. Think of them as a construction crew. You've got helicase, the demolition expert that unzips the DNA. Then there's DNA polymerase, the master builder that lays down the new strand and even proofreads its own work. Primase gets the site prepped. Topoisomerase keeps the whole thing from getting tangled up. And finally, DNA ligase comes in like a finisher to glue all the pieces together seamlessly.
Okay, we've made it to our final section. The microbe has energy, and it knows how to replicate. But the real genius is in knowing when to do things. How does it decide which genes to turn on and which to leave off? This is gene regulation. It's all about flipping the right genetic switch at exactly the right time.
So bacteria have two main ways of controlling their genes. You have inducible genes, which you can think of as motion-activated lights. They're normally off, and they only flick on when a specific molecule, an inducer, shows up. Then you have repressible genes. These are like the lights that are always on, and they only turn off when a molecule comes along and actively flips the switch. And bacteria manage these switches in this incredibly streamlined way using something called an operon. An operon is brilliant. It's a bunch of genes that all work together for one task, all lined up in a row, and controlled by a single master switch. It's like having one circuit breaker for the entire kitchen instead of a separate switch for the fridge, the toaster, and the microwave. It's just super efficient.
And here are two textbook examples of this in action. The lac operon is inducible. It's the system for digesting lactose, or milk sugar. The bacteria only turn it on if lactose is around and its favorite food, glucose, is gone. It won't waste energy making lactose digesting enzymes if there's better food available. Then you have the trip operon, which is repressible. It controls the genes for making the amino acid tryptophan. If the bacteria can just grab some tryptophan from its environment, it shuts the whole factory down. Why build something you can get for free? It's pure, simple, economic logic.
And that really brings us full circle, doesn't it? From the drugs we use to target their unique machinery, to the energy pathways they depend on, to the replication process they use to multiply, and the smart genetic switches they use to adapt and survive. Every part of their biology we've talked about is a window into their world. And in this constant arms race against evolving microbes, you have to wonder, maybe understanding their playbook, inside and out, isn't just our best defense. Maybe it's our only real path forward.